Welcome to today's daily revision booster on plant diseases, alcohols and radiation in and out of atoms. First up, today's daily formula is the formula for kinetic energy. So kinetic energy is measured in joules, and to work it out we do 0.5 times the mass in kilograms times the speed squared. In today's Biology Blast, we're going to be having a look at plant diseases. So what we're going to do to start off with is have a look at the examples you need to know for the OCR spec. So our viral disease is tobacco mosaic virus, or TMV. And when a plant actually has TMV, then what happens is it gets these mottled or discoloured leaves. And the reason for that is that the chloroplasts are not being allowed to form by the virus. If we don't have those chloroplasts present, then what happens is the plant can take in less light energy and therefore there's less photosynthesis, which means that we get stunted plant growth and lower yields. If we have plants that do have TMV, then in order to prevent the spread, we remove those infected plants and we're then going to wash any of the equipment and our hands between any plantings to reduce the spread from plant to plant. If you've also had an outbreak one year, then what you would do is plant TMV resistant crops in that area the following year just to stop it coming back. The bacterial disease in our plants, then we've got crown gall disease, which is caused by a bacteria called Agrobacterium tumefaciens. What happens when we have a plant with crown gall disease is what you can see on the right there. So you can see that we've got this big lump basically in the centre of that plant. So the actual bacteria will enter through a wound and then it integrates its plasmid into the plant genome. As a result of that, it produces a lot more of the growth chemicals and then it just causes this excessive growth within that region of the plant. So we end up with this thing called a gall. So you can see there, it might just be in one part, it might actually go all the way around the trunk or stem. And if it does that, it can cut off the flow of sap, which again can lead to that stunted growth and in extreme cases even death of the plant. In order to prevent the spread of our crown gall disease, then we take any infected plants and destroy them. We'd also avoid planting susceptible plants in that area for 2 years just to make sure the bacteria are no longer present. Finally, we have our fungal disease, which is powdery mildew. Now, when we have a plant with powdery mildew, then we end up with these white powdery spots on the leaves and the stem, hence the name. And what we'll also see is a reduced growth of our plant and the possibility that leaves drop early, which again leads to a reduced yield. In terms of how this one spreads then, because it is a fungal disease, then high humidity and moderate temperatures are really favourable to it. And it's going to survive on those plant residues between the seasons so that when the plants then grow, it can reinfect that new season. The reason it can do that is that the fungal disease itself is going to release spores which can be spread by the wind and then sit in that plant residue in the soil until the plants start to grow and then a new infection takes hold. To actually prevent this one spreading, we just spray it with a fungicide, which is a chemical that kills fungi. For those of you doing combined science, you can now jump on down to the physics prep, which is your next section for today. For those of you doing GCSE biology, then stick with me. So the next thing you need to know about to do with our plants then is all about how they can defend against disease. And we've got two types of defense mechanism here. The physical defenses, which are the physical barriers that prevent those microorganisms entering in the first place, or the chemical defenses, which are the substances that the plant secretes in order to kill the microorganisms. In terms of the physical defenses first of all, first and foremost we have this structure called the waxy cuticle. Now on the leaf you can see at the bottom left there, then that's the little line going right the way across the top surface there. So it's a waxy substance that covers the epidermal cells on most parts of the plant. It's got a couple of key features here. First of all, it reduces water loss. Secondly, it's going to prevent pathogens coming into direct contact with those epidermal cells, reducing the chance of their infection. And it also prevents water from collecting on the leaf because that wax is hydrophobic, so it repels water. So that means that the fungal spores won't germinate on the leaves and therefore are less likely to take hold on our plant. The next physical defence we've got is the cell wall itself. Now, the cell wall is a major defense against these fungal and bacterial pathogens because it's an excellent structural barrier. So all plants have what we call a primary cell wall, and that's going to provide the structural support for the cells, as we've already discussed in B1, and it's made up mainly of these cellulose fibres. So it's got the strength and the flexibility it needs. Those cellulose fibres are actually cross-linked with other substances like pectin, and it forms a gel which then helps to cement those neighbouring cells together. So what we've done there is we've formed this really good barrier against any of those pathogens. In a large number of cells, we also have a secondary cell wall which develops inside the primary cell wall as that second layer of defence. If we now consider the chemical defences that our plants have, then these ones here are the ones that you need to know. So in terms of our citronella and pine resin, then that just acts as an insect repellent, so it just keeps the insects away from the plant in the first place. The pyrethrins are insecticides, so it kills insects. Phenols are the ones that disrupt bacterial cell walls, and the defensins disrupt the cell membranes, so they're both antibacterial compounds working against bacterial cells. Chitinases break down the chitin in fungal cell walls, and caffeine is toxic to fungi and insects, so those two fall under the category of antifungal compounds working against fungal diseases. And finally, cyanide is just toxic to any living thing at all, so just something that's going to kill off anything that comes into contact with it. The last bit of today's Biology Blast is just for those of you doing higher tier biology, so foundation folks, skip on to the chemistry check. For those of you doing higher tier biology, you need to know how we can identify these plant diseases as well. And we've got two key techniques when we're looking in the field. So the first one is observation, the second is microscopy. When we say observation, then we're literally looking at those visual symptoms of the plant disease. You're looking for discoloured leaves, any signs of clear rotting, any deposits on them. So anything you can physically see on the plant, that's one of the observation techniques. Microscopy is being used to actually identify the pathogen. And what we're usually using here is a light microscope. Now, what we'll find is that's usually enough to identify it, but sometimes we may need to use electron microscopy for a more accurate diagnosis. Do remember obviously those differences between light and electron microscopy from B1 in terms of light microscopes being portable, taking them out, easy to use in those remote locations, whereas an electron microscope you certainly can't take it into a field with you. The big downside of identifying plant diseases in the field through these techniques is that we can only really do this once the infection has taken hold and the symptoms are clear to see. So our other option available to us is lab techniques to identify the disease. Two key lab techniques you need to know about: DNA analysis and antigen identification. With the DNA analysis, what we actually do is we place samples of our unknown pathogen into our little gel electrophoresis, and then we run it against known diseases. Now from that, all we do is we match up the bands. So in the case I've given you there, we can see our unknown pathogen in the middle. The bands match up perfectly with the cucumber mosaic virus, so that is our pathogen. In terms of the antigen identification, then hopefully we remember that antigens are protein spikes on the surface of the microorganism, and that specific pathogens have specific antigens. So if we identify the antigen using chemical analysis, we then identified the pathogen. And the key thing about these lab tests is that they're going to allow for the identification of a pathogen before significant damage has been created. We're not having to wait until we've got a range of visible symptoms to actually diagnose what it is, so we can get treatments in earlier and therefore hopefully have a more effective plan. Quick reminder of today's daily formula: kinetic energy in joules is 0.5 times the mass in kilograms times the speed squared. Today's chemistry check is just for those of you doing GCSE chemistry, so we're going to keep focusing on C6 and that organic chemistry topic with looking at alcohols and the carboxylic acids today. So first thing is that the alcohols form what's called a homologous series. And the reason for that is they've got that same general formula. In this case, it's CnH2n plus 1 OH. Now, they've all got that ending OH because that is the functional group. It's a hydroxyl functional group. So if ever they ask you what is the functional group in alcohols, it's hydroxyl, or you could write it as OH with the little bond as I've shown you there. When we're actually coming to name alcohols, then what we need to remember is all of their names end in OL, ol. The first part of the name is the number of carbon atoms again. So if we had 1 carbon atom, it would be methanol, 3 carbon atoms, propanol. So as long as you've learnt the first 4 carbon prefixes, so meth, eth, prop, bute, all you've got to do is add OL onto the end and you've got the name of the alcohol. If we now consider the reactions of the alcohols, then when they burn in oxygen, they will produce carbon dioxide and water in the case of complete combustion, or if we have incomplete combustion, then it's water vapour, carbon monoxide and carbon. So exactly the same products as we've seen with our alkanes and our alkenes. Another reaction we can see with our alcohols is that they can be oxidized to form the carboxylic acids. When we're referring to a carboxylic acid, this is a chemical that's got the functional group COOH. So that's the carboxyl group. So again, if they ask you what's the functional group of a carboxylic acid, you can either write carboxyl group or you could write COOH. Key thing about naming the carboxylic acids is that they will all end in anoic acid. And the first part of the name, again, just the number of carbon atoms. So 1 carbon, methanoic acid, 2 carbons, ethanoic acid, 3 carbons, propanoic acid. So all follow that exact same pattern with the naming. For their general formula, then what we've got there is CnH2n plus 1 COOH. One of the things to be really careful with here is that one of the carbons is actually in that functional group of the COOH. So do go careful not to add that extra carbon. So if you had pentanoic acid, which will have 5 carbons because pent means 5, then 4 of them are in that original carbon block, and then the 5th carbon is in the carboxyl group, COOH. So you can see the formula at the bottom there. As we've already mentioned, if we've got an alcohol and we oxidize it, then we make a carboxylic acid. So the way we oxidize our alcohol is by reacting it with an oxidizing agent like potassium manganate 7. So we'd start off with ethanol and we'd end up forming ethanoic acid, for example. So the way we'd actually do this reaction is we take our ethanol, we take our oxidizing agent of our potassium manganate 7, and we add a bit of dilute sulfuric acid. When you warm it, what we're actually doing is triggering this reaction. Now, what we will form is the ethanoic acid, but we can also see the reaction taking place because the manganate 7 ions are purple at the start, as you can see in the picture on the left there. And during this reaction, where the ethanol is being oxidized, then the manganate ions are reduced to manganese 2 ions, which are pink in colour. So that means that we can see that colour change from the purple to the pink during this reaction. When it comes to the reactions of our carboxylic acids, first thing to remember is they're weak acids. So they're partially ionised, but they are acids. So that when we're considering some of those common reactions, then they behave the same way as other acids we've encountered. So if we react a carboxylic acid with a metal, we make a salt and hydrogen. Carboxylic acid with an alkali or a base, we make a salt and water. The carboxylic acid with a carbonate, we make a salt, a water and carbon dioxide. When we come to name the salts though, this is where we get a bit of a variation from our other acids that we've encountered in other topics. Anytime our carboxylic acid is making a salt, the salt has the ending OATE. So if we have a look at the example there, ethanoic acid plus sodium carbonate will make sodium ethanoate, the salt, plus water and carbon dioxide. So just remember the ending of the salt is slightly different here and they will end in O8. Another quick reminder of today's daily formula: kinetic energy in joules is 0.5 times the mass in kilograms times the speed squared. Today's physics prep is looking at radiation in and out of atoms. So this is for physics, combined, higher and foundation, one for everyone. First thing, we hopefully remember from our earlier work that when we're considering the arrangement of the electrons, then they occupy certain energy levels around that nucleus. Now, when we think about different atoms, then they've got different energy levels for those electrons. But the electrons will occupy the lowest possible energy level. So that's the one closest to the nucleus. What we're going to look at then is something called the photon model. So in our photon model, electromagnetic radiation is emitted and absorbed as these packets of energy called photons. Now the energy of each photon is proportional to the frequency. So if we consider our structure of the atom, we know we've got these electrons in their particular energy levels. And if we want to excite an electron to a higher energy level, so one further from the nucleus, then that electron has to absorb a photon of the right energy. Now, once the photon has been absorbed by the electron, that electron moves to the higher energy level and the atom is in its excited state. When we actually come to think about what this really means, then one of the key things we get from this principle is that we can get something called an absorption spectrum. So if we pass light of all frequencies through a gas, then some frequencies will be absorbed. What we then end up with is this picture at the bottom, and this is the one for hydrogen. So what happens is you get those black bands or black lines that are the frequencies that have been absorbed by the atoms present. Now, obviously, when they absorb those photons, then the excited electrons move to the higher energy level. And what we end up with then are these gaps, if you like, in the spectrum, because they've been absorbed by electrons. So what we can actually do is we can use those bands to then identify what gases are present. There are some cases where a photon has enough energy to actually completely remove an electron from the atom. And when that happens, we've ionized the atom. So the ones that we're looking at here that have enough energy to ionize atoms are the photons of ultraviolet, X-ray and gamma ray frequencies. Once we've excited these electrons to their higher energy level, then they don't stay there forever. They will move back to the lower energy level. And as those electrons move from higher to lower energy levels, then they emit radiation. So we can then produce an emission spectrum, like the one at the bottom there, that shows the set of frequencies of radiation being emitted by an atom as those excited electrons move to the lower energy levels. So all we've got there are the actual lines of the actual frequencies being emitted in this case. The actual frequency of the radiation that's emitted when this occurs is going to depend on the difference in energy of the energy levels they're moving between. And what we find is it's not a case of it has to drop in one complete move from wherever it's ended up excited to back to its original energy level. It can actually take place in several little steps. So that if that is the case and it's occurring over two or more little stages, then the photons that are being emitted will have less energy and lower frequencies and longer wavelengths than if it was to do it in one go on its own. So when we're actually thinking about what that energy difference could be, then the absolute largest energy difference that we could see is from the energy level that's just below ionization. Obviously, if it goes above that, it's ionized and therefore it's not going to drop again. But just below that ionization level, that will be the greatest drop that we could then see. And the actual amount that that energy could be will vary between different atoms. So if we think about hydrogen, it can emit ultraviolet photons. But if we move to carbon, then that one can actually emit X-ray photons. So different atoms will be able to emit photons from a different energy level. Last thing to remember is that in our nucleus, where we find our protons and neutrons, then they also occupy energy levels within the nucleus itself. And because of that, then they can emit gamma rays. And the whole reason they can emit a gamma ray is because the energy involved is much higher in their movements. Hopefully at the end of today's Daily Booster, you can recall the actual plant diseases, how our plants defend themselves against it, and how we can identify them. You can recall key facts about the alcohols and carboxylic acids and their reactions, and you can also describe radiation moving in and out of atoms, including the energy levels of our electrons.